we are looking at the lab diagnosis of gas gangrene, gas gangrene is mainly caused by Clostridium perfringens. It's also called as Clostridium welchi. Okay, this is the most important one. The thing is, these actually produce, there are a lot of types, okay. The type A1, which produces alpha toxin, this is the one that causes the gangrene. So I just want you to focus on this one. Type A strains produces alpha toxin, okay. Type B strains are there, C, D, E, so many are there. Type A is the one that causes gas gangrene, okay, and it causes gas gangrene. It has alpha toxin. So I just wanted to add that information. Let's move on to lab diagnosis. We have already seen the clinical manifestations. Lab diagnosis, so far we have seen what specimens to collect. Microscopy, we saw direct microscopy. Then we saw culture media, if you remember. We saw the culture media, gas pack, onyx and oxomat. Then coming to identification of C. perfringens, because of this alpha toxin, I told you, right, we can identify the bacteria. Type A strains produce alpha toxins, okay. So there are many ways of identifying the Clostridium perfringens. You have target hemolysis, Nagler's reaction, reverse Camp test, heat tolerance test, and litmus milk something is there. This target hemolysis, let us look at this one now. Target hemolysis, it is also called as double zone hemolysis. Just look at this diagram, this photo here taken from Apurva Shastri. This one here, this pink one, the first one, can you see? In the middle it is little bright and surrounding it is little dull pink, right, dull. So middle it is clear, complete hemolysis, and surrounding it is dull, that is incomplete hemolysis. So this is done by what? Clostridium perfringens. It will have dual or double zone hemolysis. Who has double zone hemolysis? Clostridium perfringens will give double zone hemolysis, okay. Now, on blood agar, Clostridium perfringens produces an inner narrow zone of complete hemolysis due to theta toxin surrounded by a much wider zone of incomplete hemolysis due to alpha toxin. So, let me put it here. Inner complete hemolysis is due to theta toxin. Outer wider incomplete zone of hemolysis is because of alpha toxin. That's the one we are trying to figure out, right, in gas gangrene. Alpha toxin. So let me explain again this diagram. Inner narrow complete hemolysis because of theta toxin. Outer wider incomplete hemolysis because of alpha toxin. So let us move on then. So you should keep all these at 37 degrees 2 days, that and all I am thinking you know. Generally if we have not mentioned, no, it will be that only. 37 degree. If we have not mentioned, it will be room temperature only. 37 degrees centigrade for 2 days. So just remember, in exam you have to write this, though it is understood, you have to write 37 degrees centigrade, okay. Then, so target hemolysis we have finished. Let's move on to the next one, Nagler's reaction. Nagler's reaction, we're moving on to this one now. Nagler's reaction. Nagler's reaction, where will you see? Clostridium perfringens, don't forget that. This is the diagram you have to draw for Nagler's reaction. For hemolysis also, target hemolysis also, you can draw one nice diagram. Now let us see this diagram. Opalescence surrounding the streak line in egg yolk agar. Opalescence surrounding the streak line in egg yolk agar. Actually here they have not streaked it. Yes, here you can see the streak one diagram here. Let me explain to you. So what is it? It's opalescence surrounding the streak line in egg yolk agar. So what you do, you take Clostridium perfringens, it produces an opalescence surrounding the streak line on egg yolk agar or media containing 20% human serum. Opalescence can be inhibited by incorporating anti-alpha toxin in the medium. The test is also positive for Clostridium bifermentans, Clostridium barrati and Clostridium sordelli, because they also produce alpha toxin. So where is the opalescence? First tell me that. Did you understand where the opalescence is? This one. Here you can see opalescence, right? Opalescence. Opalescence. Opalescence is because of the Clostridium perfringens bacteria. Here you had already put an alpha and anti-alpha toxin, so there is no opalescence here. No opalescence because you had put anti-toxin. You understood, right? No opalescence. SCENCE, no opalescence due to the alpha antitoxin. Okay. Here they have tried to show opalescence and here they have tried to show no opalescence. Similar, but here they didn't streak it. Okay. But this can be produced by many other bacteria. You should know that, the alpha toxin. Then we have a reverse camp test. Reverse camp test, the diagram is like an arrow. Let me pick up the diagram from Apurva Shastri. You can draw whichever diagram you are comfortable with. This one or this one. First, understand the reaction. Reverse camp test, Clostridium perfringens is streaked over the center of blood plate agar and Streptococcus agalactiae is streaked perpendicular to it. So here they have put Streptococci vertical, Clostridium perfringens they have put horizontal. The presence of enhanced zone of hemolysis, arrow shaped, pointing towards Clostridium perfringens indicates the test is positive. Nice, na, this one? It will create an arrow towards Clostridium perfringens and say, yes, see, Clostridium perfringens is there. This is in blood agar plate, okay? Reverse camp test. Interesting, right? So, Streptococcus agalactiae, or you can say group P Streptococci, you will streak it perpendicular to Clostridium perfringens. And you should be able to see after 2 days in at 37 degrees centigrade. If you keep this blood agar plate, an arrow is formed facing Clostridium perfringens indicating a positive test. Nice, na? Reverse camp test this is. You don't need to, you can check more on this, how it happens and all that. 1 second. This is called as butterfly appearance also. Basically what happens, there is an attenuated zone of hemolysis. So this actually is an attenuated, this arrow thing is an attenuated zone of hemolysis. Okay, so we have covered target hemolysis, Nagler's reaction, reverse camp test positive. Heat tolerance also let us look at. Heat tolerance test, Clostridium perfringens can grow when Robertson cooked meat broth is incubated at 45 degrees for 4 to 6 hours. This differentiates from other organisms. So it can tolerate heat. In Robertson cooked meat, it can tolerate heat. Clostridium perfringens can tolerate heat. Then coming to litmus milk, Clostridium perfringens produces stormy clot reaction due to fermentation of lactose producing acid and vigorous gas. So it is producing a stormy clot reaction because it ferments lactose producing acid and gas. So anyways, this also you can know if you want. Okay, So let us see what and all we have covered in this video. In this video, we covered target hemolysis. So there will be double zone hemolysis. We have shown you that. Nagler's reaction, we showed you opalescence surrounding the line on streak line on egg yolk agar. Reverse camp test, that arrow thing, will be positive. Heat tolerance test will be positive because they can tolerate heat even at 45 degrees centigrade. Litmus milk, it will produce stormy clot reaction because it can ferment lactose by producing gas and acid. So 5 things we have finished in this video. Very good. See you.